episode. In that episode, we discussed about DNA and chromosomes. In this one, we are going to talk about how Thomas Hunter Morgan showed that genes are contained in chromosomes. We are also going to talk about genetic linkage. Oh, that's quite exciting. But let me answer the quiz question I gave you last episode. The C stands for, drumroll please, sound editor. Please give me a drum roll. You can't? Okay. Cytosine. Okay. But you might say you're spending a lot of time to end this cliffhanger. And I will. So let's stop beating around the bush and start this episode. Now, Morgan was initially quite skeptical of the work of Sutton and Boveri until he started doing experiments on fruit flies. Well, why fruit flies? Well, they are quite cheap and they have babies in a very short amount of time. This is good for two reasons. One, you can buy two fruit flies, one male and one female, put some food at the bottom of a jar, and in a few months, you have a ton of fruit flies. The second reason is that it's really easy to see genetic differences between generations. Okay, but enough beating around the bush. What did he observe in fruit flies? Well, one stray male fruit fly developed a mutation, a change in its genetic code that made it have white eyes instead of the usual red. And this led to a breakthrough discovery by Morgan. Let me explain. Now, before we discuss how Morgan experiments proved the Boveri-Sutton theory, we first need to discuss what the results of his experiments were. Now, he had this white-eyed male and he bred it with the red-eyed females. All the offspring were red-eyed. Now that makes sense, given that white eye is clearly a recessive gene. Okay, but what happens next? What happened when he breed these newly born red-eyed males and females? Well, he got 50% red-eyed females, 25% red-eyed males, 25% white-eyed males, and no white-eyed females. Now, what's going on? First, we need to talk about sex. Now, biological sex is simply determined by a specific pair of chromosomes. We call them the X and Y chromosomes. Now, if this pair reads XX, then it's a female. But if it reads XY, then it's a male. Okay, but why am I touching upon this? Well, allow me to illustrate with my illustrations. Now, let me quickly draw a Punnett square. Yay. Now, let's assume that genes are carried on the chromosomes. And the specific gene for eye color was carried on the X and Y chromosomes. Well, in this case, the white-eyed male will look something like X, Y. Let me just draw a W on top of the X so we know it's white. But the red-eyed females would look something like a red X and another red X. Okay, now let's have fun with Punnett squares. Now, for those in this column, we got a red X and the Y from the white-eyed male. white-eye genes. But the ones in this column, well, they have one red-eye gene and one white-eye gene. Okay, so this is a diagram, but what does it show us? Well, these ones over here, they are males which don't have any white-eye genes. They are totally normal males. But these ones here, they are not normal females because they have this white-eye gene in the middle of it. Let's call them carrier females for now. When the carrier females breed with these totally normal males, guess what happens? Okay, let me just quickly draw these normal males. Okay. Okay, so what happens when we breed these? Well, we get a totally normal male, the same male we had when we started this procedure. This is a totally normal male. Now over here we got a carrier female. Again, both of these are red-eyed females. But down here, XWY, the same white-eyed male we had when we started this. But they are no white-eyed females. Now, this is the exact same result that Morgan got. Now, this didn't 100% prove that chromosomes contain genes, but it made it much more likely. And as more and more experiments piled up on this, we finally understood how chromosomes work. Now, there's an important concept known as genetic linkage. Now, we have thousands, millions of genes in our bodies, but only 23 pairs of chromosomes. So, obviously, we'll need multiple genes on each chromosome. And this results in some wild and wacky behavior. Specifically, genetic linkage states that two genes quite close on a chromosome might be inherited together without independent assortment. Why is this? Well, if two genes are on different chromosomes, well, then obviously they'll assort independently because half of them are from your mom and half of them are your dad. There's a 50% chance that any chromosome in the parent will be inherited. But what about genes far apart on the same chromosome? Well, recombination to the rescue. Allow me to illustrate. Now let's say you have this chromosome pair. Now you have these two genes here, big A, little a, big B, little b. Now, genetic recombination states that sometimes at the beginning of the process, these pair will exchange parts of their chromosome. So, for example, this may get swapped and we'll have AB, AB. Now, this happens a lot. Now, when the two genes are far apart, this happens enough that the odds that they'll be inherited differently are very close to 50%. So for all intents and purposes, they show independently. But what happens when they are close together on the same chromosome? Well, before, a recombination swap could happen anywhere in this area. But now it has a way smaller target area for recombination to occur. So it will happen way less likely. And this means that the odds are really high that A and B will be inherited together. This is genetic linkage. Now, we can make maps on genetic linkage, measure it with centimorgans and units like that. But for all intents and purposes, this is what you need to know about genetic linkage. Now, I hope you enjoyed this series on evolution and genetics. I'll finally end it with this quiz question. Genetic linkage is an exception to which Mendelian law? Give me the answer and I'll see you in the next series